In previous class, we have discussed about the process of replication. Now, we're going to study some of the experiments that prove that DNA is the genetic material. The first experiment that we're going to discuss is Griffith Experiment. It was Frederick Griffith in 1928 had performed the experiment. He had taken a bacteria, known as Streptococcus pneumoniae. So this is a bacterium that will infect the human and they will cause pneumonia disease. So this pneumonia, Streptococcus pneumoniae, was injected in a mice. Before that, let's try to see the two type of strand. That is S strand and R. Just write S and R strand. Okay, two type of Streptococcus pneumoniae. Now this S is having a special kind of a smooth polysaccharide layer. Okay, smooth polysaccharide. Okay, coat. Whereas in the R strand they don't have. Okay, so the S strand is considered as virulent, means they are virulent, they can cause death to an organism, whereas the R strand is non-virulent. It means that they are not harmful. Okay. Now, Griffith, what he have done is that he took S strand and he injected on the mice. What happened? Since it is virulent, so mice dies. Okay. Next, he took R strand, and he injected in the mice. Mice will be alive. Okay. Why alive? Because it is non-virulent. It is not going to cause death to an organism. The next was he killed. He heated the S strand. Okay, he heated the S strand, heated and killed the S strand and injected in the mice. It means he degraded the S strand and injected on the mice. And what did he find? The mice was alive. Okay, the mice is still alive. Now, in the fourth time, he had taken the heat-killed S strand plus R strand. Then what did he observe? Okay, he observed that the mice dies. It was heat-killed S strand, which was already killed, whereas the R strand was non-virulent, but still the mice dies. So Griffith come up to a conclusion. What made the R strand to be virulent? Which factor? How the R strand, which was non-virulent, has been transformed to virulent by the S strand? Because he understood that it was S strand which made the R strand virulent. So Griffith come up to a conclusion that it may be because of the genetic material, but he did not come up with the idea that whether it was a DNA, RNA or protein. Okay, so we call Griffith experiment as transforming principle. Transforming principle. Now, based on this transforming principle, there were three scientists, Avery, McLeod and McCarthy. they have come up with a new experiment later on. Let's discuss this experiment. Now, what they have done was they have taken the est, okay, which was the S strand, heated, killed S strand, okay, and from there they have purified, okay, biochemicals, okay, they have purified biochemicals, means they have removed the lipid, okay, and they have only RNA, protein and DNA. Okay, let's see in the form of diagrammatic presentation. So this is our test tube, just say, okay, this is another test tube, this will be the third test tube. Now in this test tube we have RNA, I'll just write as R, P, protein and DNA, okay, in all the test tubes. RNA, protein and DNA, RNA, protein and DNA. So they have purified these biochemicals from the heat-kill S strand. Now what did they do was they used digestive enzymes. What is digestive enzymes? Means they are going to degrade. Okay, they are going to degrade based on the digestive enzymes that we are going to use. So in test tube 1, test tube 2 and test tube 3. In the first test tube they have added protease. So what is protease? They are going to degrade the TEASE. Protease, they are going to degrade protein. So in the second test tube they have taken RNAs, and in the third they have taken DNAs. It means that if RNase is there as a digestive enzyme, they are going to degrade RNA. DNase will degrade DNA. Now, it means that once you add protease in this test tube, there will be only protein and only RNA and DNA, right? Okay. So what they will do? They will add the R cells. Okay, they will add the R cells. R cells means the non-violent strand. Okay, same thing here. Okay, here will be, since RNA is degraded, so there will be only protein and DNA. So here also you will add R cells. Okay, in the third test tube also, where there is no DNA. So this is P and there will be only your RNA and protein, no DNA. So here also you are going to add R cells. After adding the digestive enzymes, you have seen that in protease, this is a sample where there is no protein. In this sample, test tube number 2, there is no RNA and there is no DNA in the third test tube. Test tube number 1, 2 and 3. Now what happened? After some time, they observed that here in this test tube, even when R cells was added, they have seen S cells. S cells is alive. They have seen S cells. Okay, means the heat kills S strands was observed. Here it was even in the second sample. Okay, but in the third sample, no S cells. So they have not observed. Okay, just right here, no S cells. So they did not observe the S cells where they have added DNAs. So what these DNAs have done? DNAs have degraded the DNA and there is no DNA in the sample. So this experiment had proved that the transforming principle or the transformation which had taken place, okay, in the R strand was because of DNA. It was neither RNA nor protein. But from this experiment, Avery, McLeod and McCarthy experiment, they come up to a conclusion that DNA is the genetic material that involved in the process of transformation. The next important experiment is Percy and XII experiment.